transcription? It's a formation of mRNA from DNA. Enzyme polymerase catalyzes the process. Transcription is brought about by transcriptional unit, which has promoter, structural gene, and terminator. Promoter is present at 5' end and it provides binding site for RNA polymerase. Structural gene is a site for RNA formation. Terminator, it is present at 3' end and it helps in termination of RNA process formation. DNA-dependent RNA polymerase polymerizes the new strand of RNA in a direction 5' to 3'. Let's see how the process goes for. Now check here. This is a segment of DNA. Here we find promoter, structural gene, and a terminator. Now remember, there are two strands of DNA. This is one strand. This is the other strand. One acts as a template while other is called as coding strand. The direction which we specify for promoter and terminator is related to coding strand. Coding strand is that segment of or that strand of DNA on which mRNA is not formed. Remember that coding template or coding strand is that strand on which mRNA is not formed and the direction are determined by coding sequence, coding strand. See here. For a prokaryotic, you will find RNA polymerase has sigma factor which binds to promoter and there is a rho factor which binds to terminator. So sigma factor helps in initiation while rho factor helps in termination. This is a segment of DNA. Let us consider this is a transcriptional unit having promoter, structural gene, and a terminator. RNA polymerase binds to the promoter. Once it binds to the promoter, structural gene open up, means the hydrogen bond between the base pairing breaks and the two strands separate out. Now you will find that there are two strands which are running opposite to each other. The strand which runs 3' to 5' is called as template strand on which mRNA is produced, while the strand which runs 5' to 3' is called as coding strand. On this, mRNA is not produced. But remember, direction is determined by coding strand. Now what happens here? DNA-dependent RNA polymerase polymerizes new strand of mRNA in the direction 5' to 3'. So see here, C is complemented by G, G is complemented with C. Only thing is that instead of thymine, you will find uracil forms a base pair with adenine. Remaining it is same. Most of the time, students ask me, sir, mRNA is formed on this strand by base pairing, then why this strand, the other one is called as coding strand? For this, I answer the student, see, the mRNA which is formed has a direction of 5' to 3', which is same as this strand. This is also 5' to 3'. And even this is 5' to 3'. Second, see the sequence, GCC, everything is same. Only thing is that thymine is replaced by uracil. Sorry, thymine is replaced by uracil. Getting it? Okay, this is clear with you, and that's the reason this is referred as coding strand. Once the process is completed, RNA polymerase reaches the terminator. It separates out from the promoter site. As a result, once again, the RNA molecule move out from the DNA strand, and you will find that DNA once again become double helix, and the RNA which is formed has a direction 5' to 3' and the corresponding base pairing. It's a linear molecule. Now, if it is a prokaryotic, the mRNA which is formed is polycistronic and functional mRNA, means it does not require any further processing. If it is a eukaryotic, then remember the mRNA is monocistronic, that means it can produce only one protein, and it is referred as hnRNA, heterogeneous nuclear RNA, because it will require further processing. Now, when I say further processing, means what? The mRNA in eukaryotic cell, the one which is formed, is called as primary transcripts or hnRNA. Now it wants to become mRNA, that is functional mRNA. So it has to undergo processing. Processing include, see, this is a segment. You will find that this segment has intron and exon. Now remember, for you, exon is a functional unit while introns are non-functional part of the mRNA here. So there is a process called as capping guanosine triphosphate. It's an unusual base pair. It get attached at the phi dash end. Phi dash end is considered as head. So we call this process as capping. Now what happens here? Introns are removed. Removal of intron, we call it as splicing. And you will find that poly A, adenine, poly A get added at the 3 end. We call it as tailing. So there is capping, splicing, and tailing. And because of this, you will find all the exons together. Now these exons are joined by ligase to form, see, ligase joined them to form a mRNA. This mRNA is referred as functional mRNA. Capping not only helps in becoming functional, but also protects the mRNA. Hope I'm clear with this.